Good afternoon to all. I welcome you all in GII webinar series. Genome India International is one of the leading society working on genomic sciences. Today in this GII series, we have Dr. Yasodha Ramasamy with us. She will deliver her talk on genomics in forestry. Before starting, I would like to introduce Dr. Yasodha with all our participants. Dr. Yasodha Ramasamy is a scientist G and group coordinator research at the Institute of Forest Genetics and Tree Breeding, Coimbatore. She has more than 30 years of research experience in forest biotechnology. Her expertise in the areas of tissue culture, tree tissue culture, clonal forestry, and genomics application in tree breeding and genetic resources conservation. Her contributions in applications of DNA markers in tree species need a special mention. Currently, she works on molecular breeding of eucalyptus, genomic selection in teak, and DNA marker-based gene ecological zonation in red centers. Thank you, ma'am, for accepting our invitation. Now the stage is all yours. I request you to please share your screen.
Good afternoon to all. Is the screen visible?
Yes, ma'am. It is visible.
Thank you, Dr. Maneet and Dr. Pras for the nice introduction. Again, on behalf of the Genome India International, I again welcome all the participants to this webinar on genomics in forestry. Before going into the presentation, I will just brief about the structure what I made for today's discussion. I will briefly mention about the Institute of Forest Genetics and Tree Breeding, and what are the major achievements we made in tree breeding. And then I will share 3 case studies in genomics. It includes eucalyptus, teak, and red sanders. Coming to the core mandate of this ICFRE, Institute of Forest Genetics and Tree Breeding, it is to develop new varieties, management, and silvicultural techniques to maximize productivity of natural and planted forests under different ecological considerations and changing environment. In this premise, we take up the research in tree genomics. Just introduce the trees which are unique in the sense, like when we compare with agricultural crops, the genetics of forest trees are something different. They are unique not only in their form and structure, but also in their genetic aspects. Unlike agricultural crops, they are largely undomesticated. That means mostly they are existing in the wild state. Selection and breeding tools did not change their genetic status yet. Of course, they are perennials with multiple cycles of reproductive and vegetative phases. Due to their large size, occupy considerable amount of land area. It has the impact on raising field trials and field experiments. And growth plasticity exists in the form of structure as well as function for various external influences, including climate change. And that is getting reflected in their physiological and morphological features. And forest trees are highly outcrossing in nature and express severe inbreeding depression. Heterozygous in nature, and no pure line production is possible like agricultural crops. And they take quite a long time to mature. Harvesting age is long, hence realizing the economic gains also takes quite a long time. However, they harbor quite a good amount of genetic variation for any selected traits, and it provides the advantage of high adaptive potential to the changing climate. With this background, the major applications of genomics are in the fields like DNA fingerprinting, molecular breeding. Genomic studies are taken up for population level adaptations, species comparison, functionality of different genes are being studied for conservation as well as tree improvement programs. And the tree species targeted majorly for biotech research are Papillus, Eucalyptus, and Casuarina. These are generally cultivated as pulpwood plantations in short rotation. The species like Pinus, Picea, and Quercus are mostly of temperate occurrence, and they have multiple utility. Then tropical trees, high value trees like the teak, sandalwood, and red sandals are the recent introduction into this genomic research. And likewise, if we see the genome size of these trees, they are also quite varying, and chromosome number, all those things have a lot of variations. Yeah, before going into the genomics related aspect, I would like to highlight the significant developments made in few species like eucalyptus and castorina, where tree breeding is yielding results, and it provides a platform for genomics based tree improvement. IFGTV has taken up the systematic tree improvement program for eucalyptus since 1995, and in the last 10 years we release clonal varieties for cultivation. Clones are produced through tissue culture as well as simple vegetative cuttings. We use high-tech propagation systems. This is the view of the IFGTP clones getting multiplied on large scale at a paper industry. These are the plantations which can be harvested at the age of 5 to 6 years. And the another species is Casuarina. Here, the harvesting is possible even at the age of 3 years, and farmers show more interest for cultivation of this species. Again, IFGTV has released varieties which are in routine cultivation. In this species, we generate interspecific crosses, and hybrid Casuarina clones are supplied to farmers, and the large-scale cultivation is happening in commercial nurseries. Hybrids also produce higher levels of wood per acre, like 60 to 70 tons are easily possible, and the morphological features are something different. They are highly favorable for the routine work like scaffolding or in the paper industry, and the hybrid clones are easy to root also. And this is the view of the high-tech mother-bed chamber, where we store the stock plants, where these plants can yield the cuttings for continuous propagation. Then coming to the genomics part, as it is, like I will present 3 case studies. The first one is eucalyptus. Here, to enhance the pulp yielding traits and adventitious rooting traits, we selected the parents from the high wood yielding phenotypes and generated hybrids through controlled crossing also. Here, such kind of high raised ladders were made, and the crossing is carried out. Currently, we could bring the flowering height of these trees to ground level by grafting methods. 2 different species with contrasting traits are always selected for crossing. For example, here you can see the mapping population 1, Eucalyptus camalensis into Eucalyptus terticornis. Likewise, we use Eucalyptus grandis as male parent and Eucalyptus camalensis female parent. Like that, different hybrid combinations we make. And whole genome information was available only for Eucalyptus grandis till 2020. And hence, we used a low depth coverage of about 10x and identified polymorphic SSRs. So for the species which are commercially cultivated in India, they are Eucalyptus camalensis and Eucalyptus terticornis. We generated indels and SNPs also for each genotypes, and all this information we have used for genetic linkage map development. In 3 species, we need to use the pseudo test strategy for generation of linkage maps. Here we develop the parent specific maps, we used both SSRs and SNPs, and the markers which are heterozygous for both the parents are considered for the consensus map generation. And we use the specific software called join map, which gives the option of 2-way pseudo test strategy. And this is the consensus map generated with SNP markers. And the chromosome scale assembly is available for different species of eucalyptus. Like since 2021, there are a lot of reports coming out, specifically from Australia and Brazil. And using those information, we could annotate the SNPs, whatever we have generated. And here the outer track represents the 11 different chromosomes of Eucalyptus grandis. Inner rows represent the SNPs based on their segregation pattern and their characteristics also we have analyzed. And the next case study is teak. Teak, of course, is a very familiar and popular timber, and it is a premier timber and preferred for outdoor and indoor furnitures, heavy duty construction, plywood generation, decorative veneers and carving. The wood is highly durable, resistant to different kinds of weather and other climatic elements, including chemicals. Wood gives attractive and pleasant appearance and takes good craftsmanship. Like the carpenter can work in this with this wood very easily. And India has the largest area under natural teak forest, but India imports teak worth something like 1,000 million US dollar every year, mainly from countries like Brazil, Latin American countries, and African countries, where these countries grow teak exclusively to supply to the Indian market. Being native to India, we have many different unique populations known for different wood features. Trait variations exist in height, diameter, stem form, branching pattern, flowering, and seed production. Some genotypes flower early, like at the age of 3 years, and some will take about 7-8 years. So the flowering is late, we get the good stem form. So we have a lot of choices for breeding in this species. Likewise, wood color, grain pattern, and the ratio between the softwood and hardwood also varies. So with these features, we try to select the best performing trees from the forest. And like eucalyptus and casuarina in teak, also the clonal forestry is highly successful. Currently, IMGD works on an all India coordinated project where teak clones are popularized across the country. Like we supply several lakhs of plants to the state forest departments for their plantations. So here this slide shows about the genetic analysis of natural teak population. Here this work was done by one of the Denmark team, where they used just 6 SSR markers and they have mapped the genetic diversity of all the natural teak and found that how the teak genetic resources moved to different locations over the period of time. This map indicates that India has got the maximum diversity of teak and provides the foundation that for tree breeding purposes, these populations are the ideal targets. This is the origin where the Karnataka, Kerala, and this region has the highest levels of diversity, and it moves up like that and goes via Orissa, then to Thailand, Laos, like that. The natural distribution occurs. Then accordingly, the allelic diversity also varies. Due to the paucity of the genome sequences, earlier, like the researchers were using only very few SSR markers. So we have taken up a project with the support of Department of Biotechnology, and we have sequenced the whole genome and assembled it into 317 million bases. And later from Michigan State University also a chromosome scale assembly was published in the same year in 2018. So these publications paved us way for taking advanced genome applications in this particular species. Here again, we analyzed the Indian population, specifically the popular population. Most of us may be familiar with the Nilambur or Kalakad, Shimoga, or Dandali, like that. These are the unique populations. They have the specific wood features as well as the growth pattern. So we have analyzed these populations and we have taken some of the outliers, like one from Gujarat and one from Madhya Pradesh. These two populations of Sangabad and Chota Udaipur again has specific wood value. So these were subjected into SSR genotyping. We have totally used 25 different SSRs, and we have also recorded the phenology over 3 different seasons. And this is about about the population genetic analysis there for all those 25 different populations. Here, if we see the genetic differentiation among the populations, it is quite large. The value is 0.2. Likewise, gene flow is very limited. That is why they are still existing as unique populations. And this is the population structure output from the structure software. Here, based on this only, we have identified the gene ecological zones. Peak populations from Kerala formed a separate cluster. Tamil Nadu and Karnataka formed another cluster. Central Indian populations formed the third cluster. High admixture among the Kerala populations and the Karnataka populations, especially Haliyal and Titimadi, is very visible. And in Tamil Nadu population, the admixture rate was quite low. And this gives us a lot of insights of how we should go about the breeding in peak. Of course, the another important breeding tool recently introduced for crops is genomic selection. We wanted to take the advantage of clonal propagation and embarked on genomic selection for teak. Recently, we got the project approved from the Department of Biotechnology. And to take up this work, we have also got a good assemblage of genetically divergent collections. In this case, we are trying to use this genomic prediction strategy so that early selection of candidates with the higher secondary wood formation is possible. We will be using about 400 individuals where we are planning to take up the 30x coverage, and SNP discovery is being carried out. Phenotyping for growth as well as wood quality traits we are targeting. In trees with the longer generation cycles, the genomic selection aids to reduce the breeding cycle by shortening the progeny test period, and genetic gains per year can also be increased. In trees like Pinus silvestris, reports are available that higher selection efficiencies and responses per year varied from 50 to 90%, and we can shorten the breeding cycle as 50%. These are the encouraging reports available so that we can apply this technology confidently in the tree species. And here from the populations, we take up the core samples like that. From the standing trees at the breast height, we take up the core samples, and we are using these for different analysis like sapwood to hardwood ratio, wood density, color, and we use the FTIR, the Fourier transformation infrared spectroscopy for wood chemical analysis, and these parameters will be used in the genomic selection experiment. Again, teak wood durability is governed by several secondary metabolites, flavonoids, alkaloids, terpenoids, phenones, and tannins. A catalog of widely expressed genes during secondary wood formation is made by the Brazilian group. Transcriptome factor gene families and genes coding for major enzyme pathways were also identified. Further functional characterization and marker-based selection with these genes are possible. In another project on genomic patterns for local adaptation and future climate-induced adaptations, we again got the funding support from Department of Biotechnology. Here, the evolutionary potential of populations under climate change predictions are possible. We understand the adaptive evolutionary process through these genomic information. So that we can predict the future vulnerability of these different populations, and we can take up measures for assisted migration where we know that this particular population may vanish after some time when the temperature increases or rainfall pattern is changing. So we can select the trees and move the seeds to the comfortable environments. We have taken up transcriptome analysis, examined across rainfall gradients because peak occurs in different rainfall gradients. So accordingly, we have taken up the analysis and expression variations have been studied. Higher gene counts for water deprivation and asthma protectants were observed in semi-moist and dry populations. This gives us an idea that how the local adaptations is linked with the rainfall pattern. And similar studies are available for few of the species like pinus and some of the tropical trees in Brazil. And again, coming to the use of DNA information in the timber business, it is of international in nature. A lot of import and export happens with wood products. And the global wood market demands legally sourced timber. Countries specifically like Australia, USA, and Europe legislatively specify compliance measures. They have the legal protection for it. And teak is often involved in these kind of international businesses because of its value in furniture and other products. In one case, Australia wanted to import only legally sourced teak and the timber harvest, so that the timber harvest benefits will go to the marginal farmers. So accordingly, they assisted the countries like Laos, Indonesia, Solomon Islands, Papua New Guinea, Myanmar, and Thailand to develop a database of DNA database, so that they will have the entire genetic diversity available in these populations that can be utilized for timber tracking. Here, to achieve the timber tracking process, the University of Adelaide and Double Helix Tracking Technology Singapore jointly developed a SNP database to verify the products from the field to the at the product level. It not only benefits the farmers but also small and medium enterprises involved in furniture manufacturing. This I can say is one of the direct applications of genomic data in forestry. Then coming to the third case study, it is red sanders. The scientific name is Xerocarpus santalinus. It is a highly threatened species. Like it is listed in IUCN Red List. In 2021, it is categorized as endangered under the category of A2CD, and it is protected under CITES Appendix 2, and it is a scheduled timber in India. Like scheduled timber in the sense like farmers who cultivate. For that, they have to take permission for cultivation as well as any harvest or sale is through government agencies only. But it is a very slow growing tree species, attains maturity after 40 years. And it is distributed in a very narrow zone. It is endemic to eastern Ghats of Andhra Pradesh, like Chittoor, Kadapa, Kurnool, Nellore, Prakasam districts. But it is cultivated in small packets in the states of Maharashtra, Gujarat, Tamil Nadu, Odisha, and West Bengal. And the wood is highly valued. One ton is about 25 lakhs Indian rupees. It is mostly illegally exported to countries like China, Japan, Singapore. The exact use is not known officially, but they say that it is getting used in the musical instruments or in the pharmaceutical industries like that. This species face severe threats because of illegal logging as well as grazing and other fire-related problems. And this wood takes very fine decorative carvings, being a highly prized timber. It is used in furniture manufacturing, musical instruments. Natural dye is getting extracted, used in fabrics and food products. Cosmeceuticals and skincare products are developed, and it is also believed to have role in pharmaceuticals. These kind of importance this species have, but we are not having the basic information on chromosome number or genome size. Here, under the National Biodiversity Authority funded project, we could establish the chromosome number as 20 with a genome size of about 0.78 picogram. So we wanted to develop the gene ecological zones here mainly for the seed collection purpose, so that we can encourage farmers or state forest departments for cultivation For that purpose, because there was no genomic information, we just initiated a transcriptome analysis, and this is the phylogeny to confirm its phylogenetic position, and some of the Fabaceae members were tested, and its phylogenetic position was confirmed. Of course, now whole genome assembly is also available. The EST-SSR markers were developed uh, from these, and those SSR markers were used for genotyping in natural populations. We study about 18 populations here. Again, 30 markers we have developed. And here, instead of going for fragment analysis using the genetic analyzer, here we have taken up the NGS-based SSR-seq. I find it more useful. This is comparatively cost-effective, and the number of polymorphism produced is also very high. Not only SSRs we get the SNPs also. And cost-effective, I can say that we have multiplexed about 13 SSR markers, and about 400 individuals. We could do the amplicon sequencing where we have gone up to the depth of 700x. So the data generated is also yields the best results. And this is the basic population genetics analysis we have done with those data. This project is still in progress. Maybe next year we should be able to help the department to identify the specific seed zones, and accordingly they can go for conservation as well as seed collection. Coming to the end of this presentation, there are a lot of challenges available as far as the forestry is concerned. The major reason is the lack of genomic resources, even for the important species, limited information on basic species biology aspects. And we have too many species with the varied uses, and species prioritization for advanced research is not available. And currently we have a very low number of field trials and field test results as it is. And the long duration is required to assess their phenotypic parameters. And we always face the paucity of information, because when I say that it takes 40 years for harvesting, even if we want to test at the half rotation age, we need to wait for 20 years. That is one difficulty associated with the species. And we also need the trained human resources. We need to develop databases. Financial support is also, we feel, there's a challenge, and the infrastructural needs. All these are hindering the genomic research. But if we see the advantages, genomics has a lot of scope. As I told, genomic selection can fasten the delivery of improved planting stock, and genes responsible for food formation will help in timber as well as biofuel production, where we can do gene transfer or any genetic manipulation is possible. And growth and development can also be designed, like we can make up designer trees for agroforestry purposes, where the crown architecture can be manipulated, root system can be manipulated, and biotic abiotic stress tolerance studies will also benefit us to have trees for in the climate change scenario. With this, I acknowledge the Genome India International for the chance provided to present this forestry aspects of genomics. I express the gratitude to the Department of Biotechnology as well as ICFRE for the funding support, as well as encouraging such advanced research areas. Thanks to all the GII members. Thank you.
Thank you so much, ma'am, for such a nice talk on tree genomics. Really, it was a wonderful talk, and definitely it will help students and researchers working in the field of tree genomics. You have done a lot in the field of tree genomics, right from generation of mapping population, the linkage map, and finally to genomic selection, gene discovery. We wishes you all the best for achieving your future targets also. Now this presentation is open for discussion. If anyone has any question, please raise your hand. We can take your questions one by one. In the meantime, ma'am, we have a one message on chat from Dr. M.N. Rao. M.N. Rao is a lead scientist from USDA Miami. He said that we have teak plants here in US Miami. We would be happy to share germplasm if this adds value to your research. We have more than 2,500 different tropical extractions conserved here, and these are available to research if that add values to your research. Your comments, ma'am.
Yeah, I will note the point. Like maybe the germ classes will be useful for us.
Hello.
Yes, sir. Please.
Yeah, thank you. Thank you once again, Yasodha. This is Rao from USDA Miami. I am the person who messaged that here in the chat box. First of all, it's very nice, interesting talk, and a lot of data has been generated, especially for the forest tree species, which is really very hard to work on. So myself, I did my PhD on sandalwood in India with University of Agricultural Sciences in Bangalore. So now I'm heading a group here, basically ornamental and medicinal plants. We have a lot of medicinal plants over here as well. For example, we have tulsi, we have terminalias, we have philanthus. So various plants over here for sure. Myself, I'm also working on coconuts here as well. So ours is a national database in the US. So as I said, we have conserved almost 2,500 or even more. And all these germplasms came from all around the world, from all different places. So these are really available for anybody who would want to collaborate or who would want to work on this germplasm. So the link has my email address as well. I would be very happy to get in touch with you guys and help in a way. It helps the scientific community. Thank you so much.
Thank you, sir. Thank you.
Anyone? You can ask any question.
Hello, ma'am.
Yes,
please.
Yeah, ma'am, myself, Dr. Vipul from Forest Biotechnology, NAU Navsari, Gujarat. Ma'am, since in a red sender, the wavy grain type is having the more, economical rewards, or means it is having the premium price. So is there any genomic information or any markers or any information are available with that, particularly in red sender?
Yeah, recently 2 papers published on whole genome sequencing. One is from our own group, another one is from University of Biological Sciences, like from Karnataka. There are 2 reports available. You will get a lot of information on genomics now.
Particularly on this wavy grain and straight grain type, it is possible to identify with those genomic reason? That is my question, ma'am.
Okay, I really do not know whether we can differentiate wavy grain and straight grain, because whether it is a genetically controlled factor or not is not established. No, we may need to work over it. Then only we can say.
Okay, thank you.
Yeah.
Thank you very much.
any other question? We have also available Dr. Chirapela Bhardwaj sir with us.
Yes.
Any comments from your side, sir?
No, sir, it was an excellent presentation and a great effort from her. We were just looking because she said all the difficulties of using GS into forestry, this thing. I was looking, are there any other ways to cut it down? Because this is one of the biggest challenge to forest breeders.
Yes.
But I really appreciate the efforts that you are putting to bring out this series of lectures. Thank you.
Thank you, sir. If anyone has any question, then you may please ask, or we can stop this webinar. So it's now, thank you, ma'am. Thank you once again for accepting our invitation for this really wonderful talk. I, on behalf of Genome India International, once again, thank you. Thank you so much.
Thank you very much.
Thank you.